Welcome everyone. In this video, we will gonna to discuss about the previous year questions of CUET-PG Life Science. So let's discuss about them. Question number 1. Molecular absorption spectroscopy based on electromagnetic radiation in the wavelength region of option 1 190 to 800 nanometer, option 2 190 to 1000 nanometer, option 3 1000 to 1500 nanometer, and option 4 1000 to 2000 nanometer. So as we know that the molecular absorption spectroscopy uses the electromagnetic radiations mostly between the region of 190 nanometer to 800 nanometer wavelength, and in a 190 to 800 nanometer wavelength it consists of ultraviolet as well as visible regions. So the wavelength of ultraviolet are from 190 to 400 nanometer, while for the visible light wavelength is 400 to 800 nanometer. So the correct option for this question will be option 1. Question number 2. The dye used for visualization of DNA or RNA in agarose gel is, so option 1 ethidium borate, option 2 ethidium oxide, option 3 ethidium bromide, option 4 ethidium chloride. So we know that the DNA or RNA agarose gel electrophoresis mostly use the ethidium bromide. So the correct option for this question will be option 3. Question number 3. The annealing temperature during the PCR reaction should be option 1 45 to 98 degrees Celsius, option 2 45 to 65 degrees Celsius, option 3 45 to 90 degrees Celsius, option 4 30 to 90 degrees Celsius. So we know in annealing in PCR mostly the temperature is between 45 to 65 degrees Celsius, because in annealing the primers are added to the single stranded DNA or RNA. So the temperature is between 45 to 65 degrees Celsius. So the correct option is option 2. Question number 4. Size exclusion chromatography is used to separate the proteins on the basis of option 1 size, option 2 temperature, option 3 net binding specificity, option 4 net charge. So the question is directly telling you the answer. It's a size exclusion chromatography. So the separation of proteins will usually occur according to their size. So the correct option for this question is option 1. Question number 5. Which proteins are present in neutrophil? A. Collagenase, B. Leukotrienes, C. Defensins, D. Cytokines, E. Antitrypsin. So mostly the neutrophil consists, the proteins are collagenase or defensins and antitrypsin. So the correct option for this question will be option 3, in which A, C and E are the only proteins which are present in the neutrophil. There are more different proteins which are present in neutrophil, but according to these options only these 3 proteins are consist in neutrophil. Question number 6. What are anti-isotype antibodies? Option 1. Directed against determinants of particular antigen combining site of a particular antibody. Option 2. Directed against antigenic determinants that are allele specific. Option 3. Directed against determinant present on variable region of light chain class of antibody. Or option 4. Directed against determinants present on constant region of a particular heavy or light chain class of antibody, but not on any of the others. So if you had read about the anti-isotype antibodies, this question is a direct question. So the correct option for this question will be option 4. The anti-isotype antibodies are directed against the determinants present on the constant region of a particular heavy or either light chain class of antibody, but not on any of the others. So the correct option is option 4. Question number 7. Which molecule does not take part in inflammatory response? Option 1 dopamine, option 2 prostaglandin, 4 leukotrienes. So at the time of inflammatory responses, mostly the prostaglandin, thromboxane and leukotrienes take part. But dopamine will not take part in the inflammatory responses, because we know when dopamine are released when we are in our excited phase or in the happiness, at that time dopamine are released. But in case of inflammatory responses, when any swelling or infection had occurred, only prostaglandin, thromboxane and leukotrienes will release. So the correct option for this question will be option 1 dopamine, because the question is asking that which molecule does not take part in the inflammatory response, and the answer is dopamine. Question number 8. Varicella zoster vaccine is administered for option 1 hepatitis B virus, option 2 smallpox virus, option 3 chickenpox virus, option 4 polio virus. So the correct option for this question is option 3. Varicella zoster vaccine is mostly given when the person is infected from the chickenpox virus, and that zoster vaccine mostly reduces your risk of getting shingles. And chickenpox virus is a disease when someone is getting blisters on their skin, or the painful rashes, or a infection is spreading on all over their skin. So the varicella zoster vaccine is mostly given to treat the chickenpox virus. Question number 9. Which microbe is not capable of degrading petroleum hydrocarbons? Option 1 Candida, option 2 Acromobacter, option 3 Pseudomonas, option 4 Aspergillus. So this question is asking which microbe do not capable of degrading petroleum hydrocarbons. So we know, if you had read about that what are the microbes that are using for degrading petroleum hydrocarbons, it is mostly in the process of bioremediation. The microbes are Candida, Pseudomonas, Aspergillus, and there are many more microbes which usually degrade the petroleum hydrocarbons, and they are used in the bioremediation process too. But only the Acromobacter will not take part in degrading petroleum hydrocarbons. So the correct option for this question will be option 2. Question number 10. Prokaryotic mRNAs contain a conserved polypurine tract. Consequences: AGGAGG, located about 7 nucleotides upstream from AUG initiation codon. This conserved hexamer is called option 1 Kozak sequence, option 2 Sine Dalgarno sequence, option 3 consensus sequence, option 4 Tata box sequence. So the correct option for this question is option 2. Question number 11. Which type of enzyme is known to remove methyl or ethyl groups from guanine bases that have been mutagenized by alkylating agents? So option 1 are photolyse, option 2 DNA and glycosylase, option 3 alkyl transferase, option 4 AP endonuclease. So the correct option for this question is option 3 alkyl transferase. As you read in the name also suggests alkyl transferase means something related to alkyl, either methyl or ethyl or any group of alkyl. They will remove the guanine bases which are being mutagenized by the alkylating agents, and the correct answer of that enzyme is alkyl transferase. And their name also suggests that transferase means it will transfer the or remove the particular alkyl group. So option 3 is the right answer for this question. Question number 12. Name the technique Matthew Meselson and Franklin Stell used to evidence that DNA replication is semi-conservative. Option 1 calcium chloride density gradient centrifugation, option 2 2D gel electrophoresis, option 3 mass DNA sequencing. If you had read about who had given the hypothesis or theory for the semi-conservative DNA replication? Then it was the Watson and Crick, which and their model shows that DNA replication is a semi-conservative process. Now that hypothesis was proven by Matthew Michelson and Franklin Stell, and they used the technique of cesium chloride density gradient centrifugation on the bacteria called E. coli. So the correct option for this question is option 1. Question number 13. Two highly inbred strains of mice, one with black fur and other with grey fur, were crossed, and all of the F1 offsprings had black fur. Predict the phenotypic outcome of intercrossing the F1 offsprings. Option 1, all have black fur. Option 2, all have grey fur. Option 3, 2 black fur and 2 grey fur. Option 4, 3 black fur and 1 grey fur. Now, when we cross the black fur with grey fur mouse, then we will get the combination of black fur and grey fur uh, mouse in their genotype. Okay, but the phenotype will be black fur, and which is black fur is a dominant trait. Okay, now when we intercross that black fur mice with black fur which have a allele combination of capital B small b cross with capital B small b. Now what will the outcome will come? Either capital B when means that the capital B have will be a black fur mice. Now or will what will have that capital B and small b. Now what they will give you the phenotypic also black mice. Now when small b and small b will cross, then it will be give a grey fur mice, which is a recessive trait. Now we at that when we cross the F1 offsprings, we will get the according to phenotypic ratio 3:1, means 3 black fur mice and 1 grey fur mice. But according to genotypic ratio, it will be 1:2:1, because 1 we have homozygous dominant capital B, and 2 we will have heterozygous alleles, which means one capital B and one small b, but it will give a black fur mice too. Okay, don't get confused in that. And last is small b, which is homozygous recessive according to genotype. So according to genotype, if question had may change according to genotype, it may had asked, then the option will be 1:1, but it is not right now asking the phenotypic ratio, so it will be 3:1, in which 3 will be black fur mice and 1 will be the grey fur mice. So please don't get confused in this crosses, and the correct option for this question is option 4. Question number 14. How many number of autosomal linkage groups have human somatic cells have? Option 1 23, option 2 22, option 3 24, option 4 1. When we talk about the linkage groups in human, then it is total of 23 in case of females and 24 in case of males. Now, in female, when it have a 23 linkage group, they have 22 pair of autosomes or a autosomal linkage group, while one is a homologous sex chromosome linkage. So in female, there is 22 pairs of autosomal linkage. Now when we talk about the male, they have the total 24 linkage groups, because they have 22 autosomal linkage groups. Now they have extra one, which is because of X or Y. So both X and Y are not homologous, so they will have extra 1 plus 1, so it's 2. So they have 2 sex chromosome linkage. That's why the male have 24 total linkage groups. But the question is asking the autosomal linkage group in the human somatic cells. So either in female or male, in both of them, the total 22 pairs of autosomal linkage groups are present. Okay, so the correct option for this question will be option 2. Question number 15. in DNA microarray techniques consist of option 1 large number of double strand DNA, option 2 large number of single strand DNA, option 3 small number of single strand DNA, or option 4 large number of double strand RNA. So if you had read about the microarray technique, then this question will be the easiest for you to solve, because the DNA microarray technique consists of a single stranded DNA. So we will select the option according to that. So it's mostly in two or three reason. So two or three option either. Okay. Now, and that the difference are large number or a small number. So DNA microarray technique consists of a small number of a single stranded DNA which will attach with a large number of a DNA sequences. So the correct option for this question is option 3. This correct option. Okay. Question number 16. Small circular DNAs such as plasmid and small viral DNAs, when have no breaks in either strand, are referred as option 1 nucleosome, option 2 closed circular DNA, option 3 nicked DNA, option 4 chromatid. So the correct answer for this question is option 2 closed circular DNA, and that's a plasmid or small viral DNA. Okay. Question number 17. Gene expression cannot be analyzed using this technique. Option 1 DNA fingerprinting, option 2 fluorescence in situ hybridization, option 3 RT-PCR, option 4 DNA microarray. So if you know what are the different methods for analyzing a gene expression, and they are fluorescence in situ hybridization we use for the gene expression analysis, we also use RT-PCR for gene expression analysis, and we also use the DNA microarray gene expression tech for gene expressions. Now only the one option DNA fingerprinting, this method is not used for the gene expression analysis. Okay, so the correct option for this question is option 1. Question number 18. The complete sequence of the human genome was published in option 1 2020, option 2 2003, option 3 2006, option 4 2010. So the correct answer for this question is option 2 2003. In that year the complete sequence of human genome was published, and that the starting year of human genome work was 1990, and the end of that work is on 2003, and at that time the full human complete sequence of human genome was being published. Question number 19. Eukaryotes have two major chromatin proteins in SMC family. They are option 1 integrin and laminin, option 2 cohesins and condensin, option 3 hemoglobin and myoglobin, option 4 actin and myosin. So according to example or the option also, you can identify it very easily, because we know actin and myosin are the proteins which are mostly in our muscle contraction level. Hemoglobin are red blood cell proteins, and myoglobin are the muscle protein. Now integrin and will take part in inflammation or any outer response, body immune response. Okay, now only cohesins and condensins you had read in the cell division also, that they will are the major chromatin proteins in the SMC family. So the correct answer for this question is option 2 cohesins and condensins. Question number 20. In general, a carbohydrate molecule with n chiral center can have. So it's a direct question. If you had read about the carbohydrate, then you will easily give that answer. And that the correct answer for this question is 2 is to power n stereoisomers. So when we calculate the carbohydrate chiral center, we have to use that to the power n formula. Okay, so the correct answer is Option 4.